Good morning, everyone, and welcome to my presentation on the next generation genomic analysis of achondroplasia in White Pekin ducks. My name is Paige Richards, and I'm a senior biology major here at Stockton University in Galloway, New Jersey. To begin, my research on this project was conducted back in the spring of 2019, where this idea really stemmed off of previous research that my faculty advisor, Dr. Barbato, was able to conduct back in 2018. Through his previous study, he was able to identify an achondroplastic phenotype in White Pekin ducks. He found this to be an autosomal dominant mutation, but aside from this novel discovery, there wasn't much else known. So my project was really able to build off of that previous idea and locate specific areas on the different genomes that we sequenced that we can compare the achondroplastic phenotype to the wild type, and in those, the areas that identify differences, those can be used in future research to identify the significance of those areas and how they could possibly play into this achondroplastic variant. My abstract is listed here to the left. I'm not going to read the whole thing, but just a brief summary of it is from the different genomic analysis that I did, I was able to identify two different types of differences between the wild type and the achondroplastic phenotype. So the first type of difference is if one of these genomes was showing expression between a certain base pair location, but the other genome had no expression. And on another note, the second type of difference I noticed is if there is more reads or more expression in a specific area compared to the other type of genome. Lastly, this research really tried to identify FGF homologies because a lot is known in FGF mutations in humans and canines. So hopefully, if we are able to identify FGF homologies in these ducks, we can then apply this to other mechanisms that are already known and use this in future research. Overall, this was just a great experience, and by the end of that semester, I was able to present this research at the Stockton University NAMS Research Symposium. And yes, it was just an overall great experience, and I look forward to going through this presentation. Okay, so just to give a little background information about previous research, my faculty advisor, Dr. Barbato, actually works as a geneticist for this outside company that essentially makes commercial stocks of ducks. And while he was working there, he noticed a characteristic phenotype that it's a shortened shank length of the duck. So as you can see in this picture to the right of a duck, the shank is essentially the leg portion of the duck between its body and before its foot starts. So he characterized this phenotype as like a dwarf phenotype, and through all the different studies that he did, he was able to show that this was an autosomal dominant mutation. And with the research that he did, he was able to characterize that this relationship between the gene was not affected by either body weight of the duck, so either how much they were eating, how much those ducks weighed during when they were growing up in the facility, or the sex of the duck. So it didn't really have any difference between the male and females. So seeing Dr. Barbato's previous research and being able to identify this as an autosomal dominant mutation that did not have any link to the body weight or the sex of the duck, this project that I worked on then intended to have like three main goals. So from these, of the two like pureline families that they were able to use, we really wanted to sequence the genomes of them, so the genomes of these wild type ducks, and then the ones that are showing this achondroplastic mutant or achondroplastic phenotype, then identify the differences between the two genomes and see like where locate any like base pairs or specific regions that either the genomes differ or that there's a different amount of expression, and then finally search for FGF4 or 3 homologies. So it was important to search for these growth factors because in previous research, dominant mutations of these FGF factors can cause chondrodystrophy in both humans and canines. So a classic example of you would see of this in canines would be like a dachshund that has shortened legs, and that's just like a maldevelopment of bone structure. So as you can see, a picture of a dachshund is located to the right. So moving on to methods and materials, this genome DNA was collected and pooled from 2 different pureline families at the facility. So they were able to separate them into 2 different stocks, one with the achondroplastic phenotype and one just wild-type ducks. From there, these genomes were sequenced using a machine called the Illumina NextSeq 75PE. Thank you to North Carolina State University that was able to do this for us with their awesome machinery. Once we were sent that data over, we then used a software called Galaxy, where these genomes were assembled against both builds of the Pekin Duck that is on the NCBI website. So that's just a standard of what they usually term the wild-type duck genome to look like. And then finally, using genomic analytics software such as Integrative Genomics Viewer, IGV, or Genomic Workbench, we are able to compare the first 3 chromosomes of these 2 different families of the short and long phenotypes seen. Continuing with methods and materials, I'm now going to go over the different techniques that I use to identify the differences in the sequences of the short and long phenotypes. So the first technique that we used, or that I analyzed, was analyzing coverage graphs. So these graphs really are just able to plot how many times the sequence is read at a specific location along that genomic scaffold. So you'll see later in the presentation, I'll show you a picture of a coverage graph, and they kind of just, I guess it looks like a bar graph a little bit, but essentially the more reads that are at a specific location, the higher the bar will be, and that indicates how much genetic information is located at a specific spot. Next, I used graphical sequence views, so that puts them in the order of the different base pairs and their location on the different chromosomes. So once I'm able to find a location that has either a lot of reads or no reads, I'm able to look at that exact location on the chromosome and identify between what base pairs this difference is noticed at. And lastly, the technique I used to identify the FGF4 and FGF3 homologies, I was looking for certain KB sequences, so I have them listed, and then I was just able to again compare these all to the NCBI reference genomes. So lastly, for methods and materials, just to finalize the technique section, when I refer to things later on in the presentation, I'm just going to explain them real quick. So when I'm referring to like wild-type genomes, they're often seen as long 1 or long 2, and then achondroplastic genomes are referred to as short 1 and short 2. The differences that I'm really looking for in this genomic data is I'm looking for specific locations where either the long or short genomes are missing some information while the other genomes showed that information, or vice versa. I'm also looking at these 4 genomes for information at a specific location if there were fewer reads displayed in a specific variant compared to the other phenotype. So if the short genome had only like 2 reads for something, but a long genome had about 13, even though that they still had some expression, the reduced amount of expression could still be like a key factor to look for as well. So now into the results section, I'm going to first start going over this table that I was able to generate by finding differences between the different genomes. So this table is really able to show those differences between the short and long genomes when I was analyzing those graphical sequence views on Genomic Workbench. The differences listed in the table are separated not only by chromosome that they were found on, but it shows how many reads of genetic information are present between specific base pair locations on the scaffold. So the table really is able to encompass the different differences I was describing to you before. So first difference I was looking for again is if one genome, a short or a long genome, was able to show expression between a base pair location, but the other genome did not have any reads for that information. So a good example of this would be in the red highlighted boxes. So for example, between the base pairs of 1345 and 1368, the long genome had expression, but the short genome did not have any reads for that location. The other difference that I described earlier is if I see a reduced amount of expression in a specific location. So an example of this would be found in the blue box, where between the base pairs of 1469 and 1544, there is an increase in expression in the short genome, but a reduced expression in the long genome. These are all just important key areas to note that could maybe be linked to this achondroplastic phenotype. And then lastly, I know I had described earlier that I analyzed chromosomes 1 through 3. Chromosome 1 is not listed in this table because when going through that data, there was no notable differences that I was able to record, so I left it out of this table. So this is just an example of the graphical sequence view that I had described earlier. This is the same instance I described back in the table from the last slide. So this is a picture of the third chromosome between the base pairs of 838 and 865 that's indicated in the red box. This just goes to show this is an example of one of those instances I described before where the short genomes 1 and 2 do not show any information or do not show any reads between these two base pairs. However, the long genomes do have reads for these base pairs. Now figure 2 is able to show a picture of the coverage graphs I was describing earlier. This is a picture of the third chromosome. So just from far away, if you look at the shape of the genomes, they all look pretty similar. There's no real areas on this graph from far away that would show much concern, but then again, as you go in closer looking at the different genomes using those the graphical sequence views that I showed you earlier, that's how I'm able to identify like specific base pair locations. However, I was able to use these later on when I was trying to identify the FGF homologies using the Genomic Workbench, because I'm able to search up specific areas to find these KB sequences on the entire genome. So I was able to find the FGF4 homologies of the 2 KB sequences at these specific base pair locations of 1021 and also 236. After the presentation of all that data, you may be wondering, okay, so what's the point of this, or like why does this all matter? So right now in the discussion portion, I'm really going to emphasize why these two types of differences that I identified before are really important. Again, the types of differences were whether there was information either present in both the short sequences or missing in both the long sequences or vice versa. Additionally, or if there was a difference in the amount of expression of certain genes for a specific location on those genomes between the short and long. Identifying just these two different types of differences is really important because, again, phenotype is not only dictated by the amount of what genes are present, but also which ones are expressed. So just different versions of whether the gene is completely absent or if the gene is less expressed could give indication as to these different areas that I identified, if they have any influence on causing this achondroplastic variant. Additionally, the presence of the KB sequences was really important as well, because identifying this indicates that this duck genome has homology with mammalian and chicken FGF4s. I know I had mentioned canines before, but that was just in the case of mutations in the FGF factors of mammalians, and canines have been very well studied, but specifically for FGF4, it's been very well studied in mammalian mammalian genomes, and also chicken. So just having these key model organisms that have been very studied before and knowing the mechanisms of how achondroplasia works in them, hopefully this can also give direction to future research of how this mechanism works within ducks. So, in conclusion, based on previous research, there was no physical differences in sex or body weight that's associated with this autosomal dominant achondroplasia that is seen in white Pekin ducks. And again, that was identified by my faculty advisor, Dr. Barbato, in his previous research. Now, through the research that I was able to do, these genomic analyses were able to identify possible locations where these variants differ. So knowing these possible locations is really able to help in future research or gives future direction to possible questions to ask as to, okay, due to time constraints, we were only able to identify differences over the 3 chromosomes of the ducks. Ducks actually have 80 chromosomes, so maybe just having a larger sample size or investigating other chromosomes could be a future direction, but also just moving on to the next step would be really investigating what role, if any of these differences that I identified, what role they play in this achondroplastic variant. Just because these differences are identified does not mean that they're all linked to the achondroplastic variant, but it narrows down possibilities or specific target areas to look at that could show these differences or that could be linked to what's causing this achondroplastic variant in the white Pekin ducks. So thank you so much for listening to my presentation. This was honestly a great experience. I was so glad that I was able to not only present this work at the NAMS Research Symposium, but that I'm also able to just continue to present this research again. I just want to acknowledge and express my sincerest gratitude to not only the School of Natural Sciences and Mathematics, which is NAMS, and all their faculty, but specifically to Dr. Barbato, my faculty advisor, for providing me this opportunity to join such an awesome research project. Not only has it allowed me to further my studies in biology, but just as a dedicated professor, you always have provided me patience, guidance, and encouragement while you continue to support me through my undergraduate career. It's been a great experience, and I'm so thankful for this. I've also listed all my references below. Okay, thank you. Have a good day.